This prism has at least 2 strands, but nearly infinite possibilities. It used to be said that every person's DNA was a unique fingerprint, but now it turns out that just like our fingers, a person can have more than one unique DNA sequence, or rather they can have several unique DNA mutations in parts of their body, and some of the same genetic mutations can occur in unrelated people. This finding was made by Scott Williams of the Institute for Quantitative Biomedical Sciences at Dartmouth's Geisel School of Medicine. Joining us to talk about his findings is Dr. Scott Williams. Welcome, Scott.
Thank you, and good afternoon.
Good afternoon. What are the key stages or opportunities for gene mutation in the body?
Well, I think the initial assumption to a very large extent was that they occurred in the germline, basically in what's passed on from parents to offspring, with a few well-defined exceptions. But in reality, every time a cell divides, there's the possibility for DNA mutation because the DNA has to be replicated to give rise to the right number of strands of DNA in the daughter cells.
So even when we talk about genes, nature, nurture, the answer becomes yes.
Yeah, in essence, yes. There's so many, the complexity is overwhelming at some level, but clearly the assumptions that we made, which you stated in your introduction about the uniformity across individuals, is not completely true. But we've known that for a while. Cancer clearly is due to mutations in certain cells, and then they kind of expand to form a tumor with carrying that particular mutation or set of mutations that are causing cancer.
Now, are these mutations like variations in a classical composition where we can still hear the major musical themes, but then there is just a little bit of offshoot, or are these mutations relatively significant sometimes?
Well, I think probably, although we don't know for sure, the vast majority of them are not going to be significant. They're not going to change anything about an individual, change susceptibility to disease. However, as I pointed out with cancer, there are clear exceptions. And, more recently, as people start interrogating this, we're seeing that a wider variety of diseases are impacted by these so-called somatic mutations that only occur in certain tissues or certain cells in their lineages, their descendant lineages. But again, I think you're right. The vast majority of what we see as it changes, either the cells die because the cells are not viable with those mutations, or they have a minimal effect on any given trait.
Now, do we only see changes or mutations due to some kind of trauma or disease, or is it a natural condition that we'll begin to see mutations in different DNA strands throughout the life?
I actually think it's the latter. We don't have a lot of really good data on it, although people have, over the past number of years, collected data suggesting that there are some systematic changes with aging. I think we're going to see more and more of that, and that's probably due to the fact that the technology allows us to look at these kinds of things in much more detail now than was possible 5 or 10 years ago.
And I think that's probably one of the key elements to this study and probably your field in general. Since 10 years ago, our understanding of genetics, our understanding of the human genome has expanded exponentially, and our tools for looking at it has expanded equally so. Is that what allowed you to conduct this experiment and make these findings? Yeah,
in essence. So what we did was what's called whole genome sequencing, although the particular paper you're referring to only talks about the mitochondrial DNA, one independent part of the DNA. But in essence, we now have a technology that allows us to sequence an entire individual, all the nucleotides in an entire individual, for just a matter of $2,000 to $3,000 or $4,000, when, of course, the first genome was orders of magnitude more than that. So we can actually look at this now in a way that was just inconceivable a decade ago, in a regular, systematic way.
So in practical terms, how does this change our testing when we are talking about, say, paternity or crime analysis or things of that nature? Is this a significant difference in how we should look or how many samples we need?
I think for the most part, it's not going to be because the number of changes, we saw, we presented the mitochondrial DNA, which is about 16,000 nucleotides long, and saw 20-point mutations across two individuals who were heavily sequenced in multiple tissues. But, even if we extrapolate that to the full genome, what we consider, if we take any given tissue, it's going to be a pretty accurate representation, although not an exact one. And I think the level of precision is going to be a little bit, is a little up in the air now, I guess is the best way of putting it. I mean, we know that there are changes. We knew there were changes based on cancer. We now know there were changes based on other diseases. But these are rare events. So they're not things that, if you're looking for paternity and you're looking at a set number of markers, you're going to see something. Or somebody pointed out in one issue, I think I was talking to someone, and they said, well, how many people have been wrongly convicted because of this? And using forensics, it might be more likely how many people have been wrongly acquitted. Because if you get a change, it will not look like the person if there's a change in one part of the body versus another.
So what you're telling me, at least in part, is all those people who wind up going to those horrible talk shows to find out whose baby's mama is whom, they're not off the hook.
They're pretty much off the hook. The likelihood is very, very small. If we're talking about, just hypothetically, a mutation every million nucleotides or every 10 million nucleotides out of 3 billion that exist in the human genome, what's the likelihood you're going to hit one that's going to be wrong if you're only sampling a small part of the genome? It's pretty small.
Going into the research itself, what is the chance that we've just gotten a series of false positives and that you happen to look at bad genes? How many samples did you look through? How wide a catalog, a universe of different genetic possibilities were you looking at?
Well, in this particular paper, we looked at the entire mitochondrial DNA molecule, which is 16,000 base pairs. The thing that's interesting about the mitochondrial is that unlike the nuclear genome, which we have one copy of the genome from our mothers and one from our fathers, we only get the mitochondrial DNA from the maternal lineage, but we get multiple copies. And the mitochondrial DNA replicates independent of the cells dividing. So you can have certain tissues, because the mitochondria is necessary as a major energy source in the cell, certain tissues have lots and lots of copies. For example, muscle has a very high number of mitochondrial DNA genomes because it's necessary to generate the proteins that generate the energy source for the cells. And we looked at everything there. Now, what we did is we sequenced fully from autopsy samples in 2 people, 10 common tissues. We actually sequenced more, but we only talk about the ones that were sequenced in both people. And for those people, we have what's called very high copy coverage or very high fold coverage, which means for each molecule they have, we were reading that molecule thousands of times. So we were pretty confident of the accuracy of the reads. Now, what we looked at in this particular case is that if, for example, you have 1,000 copies of the mitochondrial DNA in a given cell or a given tissue on average, we asked how many copies of that mitochondrial DNA were different than the majority. And that's called heteroplasmy.
And in looking at those 2 people, is that enough for us to be able to generalize to the population? Are you worried that there is a chance that those 2 people were outliers?
I would have been with perhaps the following 2 exceptions. Some data of this kind was already in the public domain. It had been published by another group at Johns Hopkins a couple of years earlier, and we went back and looked at their data. And shockingly, they found exactly the same mutations in the same points, but only in the same tissues as we did. And then we looked at one of the mutations in 2 more people and found the same pattern. So actually, for at least some of them, we had 4 up to 6 replicates. And the results were amazingly clean, such that I think there is little doubt that these were false positives. I think this is, independent labs multiple unrelated individuals are seeing the same kind of tissue specificity, where these particular mutations only occurred in specific tissues. That was the real shocker, not that we were seeing changes per se, but that we're seeing these kinds of things occurring in a tissue-specific manner.
It really is fascinating. It makes me also wonder, there's a trend towards personalized medicines, and I'm wondering if just knowing this, that there is mutations even within that person, adds to our ability, or might potentially in the future add to our ability to make much more specific and effective cures.
Yeah, I think it probably will, and certainly there are, other than cancer now that we're beginning to see, other diseases, neurofibromatosis, some atrial fibrillation, certain actually neuropsychiatric disorders are due to a small or moderate percentage of cells in an organ having a mutation. So I think that we're not going to be able to do what we've done as geneticists, is draw some blood, take the DNA out, and assume that that's a representation of the entire genome. As we talked about earlier, on average it will be. But those few exceptions may cause diseases in specific tissues that are going to require a little bit more care.
And when we talk about the efficacy of treatment, often we're talking about certain time windows. So perhaps by being able to do these analysis of the genome, you'll be able to be more accurate as to what stage someone is in with certain conditions, and thus even be able to form a better treatment schedule or plan.
That's always the hope. I think that, as a geneticist, I think we're far away from a lot of that, but probably in pharmacogenetics, looking at treatment, we may be closer. But I think that's where the field's going, and that's what we're looking to do.
And what are you looking to do next? What's your next step?
Well, what we've done so far is published just the mitochondrial. We are actually examining the data from these same individuals now, looking at the whole genome, and I think that's going to be a fascinating question. And then ultimately, the question is, if we accumulate mutations as we age, can we use those as markers of disease risk in a tissue-specific way? And I think that's a long-term objective. But probably the shortest-term objective is going to be looking at that in cancer, and we're going to, and trying to see whether or not we can use variation across the body and compare it to cancer as a means of identifying what's really driving cancers in particular tissues.
In some respects, it really does make sense. I mean, everything about us, as you said, it changes. Day to day, year to year, decade to decade. So why wouldn't there be, as those cells split, changes potentially at that level as well?
Yeah, and one of the things that we've thought about, although we haven't been able to do it, is if you're a smoker or a drinker, do your lungs and liver mutate more?
Actually, yeah, I was thinking about that when I did the nature-nurture bit, that it's very likely.
Yes, it is. I mean, but getting that data is going to be really difficult because it's hard to get the right tissues. I mean, we had to, what I didn't specify, but in that study, we specified we didn't want anyone who had cancer.
Are you going to, or have you done, or has anyone done, to your knowledge, a longitudinal study of this sort, where you could look at tissue samples at someone at 6 months, at 5 years, at 20 years, eventually, obviously not at 60, the science is too young, but would that be of value?
Oh, yeah. The problem is that it's almost impossible to do that in a given individual. So what people have done is, especially for mitochondrial variants, they've looked at, let's say, muscles as people in different age groups to see whether or not specific mutations accumulate. And the answer is they do. So, you can look at people who are, 20, 40, 60, 80, sequence it, but the studies that have been done up till now are looking for one or two specific variants. We had the ability to look across the entire genome in one shot of the mitochondrial DNA, and that was really allowed us to kind of generalize.
Yeah, and isn't it amazing that we can do that?
It is, yeah. I mean, it's amazing. And when I quoted the prices are actually cheaper. I just don't want to annoy my suppliers. I mean, you can do a whole genome now for $2,000 to $3,000.
Looking through the prism to discover that you need more than just a thumbprint to understand someone's DNA. I'm Andrew Hiller for The Voice of Russia. And I've been joined by Dr. Scott Williams of the Institute for Quantitative Biomedical Sciences at Dartmouth's Geisel School of Medicine. Thank you so much for your work and your time.
Thank you very much.